はい。皆さん聞こえておりますでしょうか。では時間になりましたので、エリックスのセッションを、始めさせていただければと、思います。で、スケジュールはですね、このような順序になってまして、まず、私が、最初にエリックスの会社紹介みたいなところでしたり、AI 創薬の割とジェネラルな、お話をしまして、で、その後ですね、弊社の、エンジニア研究者の方からもう少し具体的な、お話、予測モデルでしたり、生成モデル、あとは逆合成解析の、お話の、ご紹介をできればと思っております。で、弊社のエンジニア研究者のセッションは英語のセッションに、なっております。なんですけれども、質問はですね、英語でももちろん全く問題ないですし、それが一番助かりますけれども、日本語でご質問いただいてもですね、私の方で頑張って翻訳して、エンジニアに伝えてみたいと思いますので、そのような形で進められればと、思います。でご質問があるときにはですね、途中でチャットボックスにご入力いただければと、思います。はい。では、始めたいと思います。でですね、まずちょっと最初に簡単に会社の、概要をご紹介できればと思うんですけれども、まずですね、僕らはですね、大体4年くらい前に設立した、会社です。で、エリックスという会社でして、私がですね、コーファウンダーでCEO をしております。で、場所は東京にあってですね、オフィスはこんな感じになってまして、で、メンバーがですね、今、業務委託とかも含めると、19名っていうような形で、去年も CBI 学会で発表させていただいたんですけども、その時は確か半分以下くらいの人数しかいなかったのかなと思っていますので、急速に人が増えているなというような、状況です。で、僕らはですね、AI スタートアップでAI を軸にビジネスをしてますというところで、最近特に一番力を入れているのが、AI 創薬のところですと。で、あとは、化合物を扱う場合にはですね、マテリアルズインフォマティクスという材料を扱うような分野でも、非常に同じような話が出てきますので、そういったところも僕らのところで扱っておりますというようなところです。あとはコンピュータービジョンという画像関係のところになりますというところです。で、最初にですね、僕らがなぜこの分野に来たのかみたいな話からちょっと入れればと思うんですけども、AI、ディープラーニングって、今本当にいろんな領域で役に立つようになってるんですけれども、一番最初に役に立つようになったのって、基本的に画像の領域なんですね。コンピュータービジョンと呼ばれるところです。で、画像の中でも本当に様々な、アプリケーションがあるんですけれども、その中でもですね、特にインパクトが大きいのが、自動運転ですとか、あとはADAS と呼ばれる運転をアシストするような機能を、扱っているところに、なりますと。で、特にあとは日本はですね、自動車産業が大きいっていうところからしてもですね、非常にインパクトが大きいところだと、思っていますと。で、今はですね、その後、だんだんそのコンピュータービジョン以外のところでも非常に AI が役立つようになってきていてですね、いるんですけれども、相変わらずですね、コンピュータービジョンだったり、自動運転だったりっていうところは、AI の中でも一番発展しているところの一つというようなところになっております。そこで、そういった、AI の、最新のその技術を、創薬に、どんどん持ってきましょうっていうところが、僕らのやっていることになります。で、ちょっとだけコンピュータービジョンの、話もできればと思うんですけれども、ですね、コロナになってからですね、僕らソーシャルディスタンシングをモニタリングするっていうようなツールをちょっと作りまして、それもですね、日経新聞さんですとか、テッククランチさん、ヤフーニュースさんですとか、そういったところに取り上げていただいたっていうことが、ございました。で、どんな風に見えるかっていうところなんですけども、こんな風になってまして、カメラの画像があってですね、そこから、こういったこうボックスをつけてですね、人をこう検出します。で、それぞれの、人のこの距離を、測ってみたいなことを、やっています。で、これ、斜めの、画像で距離を測ると、こう、正確な距離にならないので、一回その、なんかバードアイビューっていう、なんか真上から見た、図に変換してやって、距離を測るとか、やっていたりしますと。 であとは、プライバシーの問題とかも最近非常にあるので、こういった今、緑色に隠されてますけども、こういったセグメンテーションのマスクなんかもつけたりっていうこともやっています。で、これ、アプリケーションとしてはですね、自動運転とかと、全然違うものには見えると思うんですけれども、自動運転もですね、非常にいろんなサブタスクで構成されていますと。なんか全部やってくれる AI みたいなのがあって終わりみたいなことではなくてですね、なんかいろんなサブタスクに分かれてるんですね。で、その中でも一番、大事なタスクの一つは、歩行者を検出するっていうようなタスクなんですよね。で、そういった、背景技術と、こういったものって、実はほとんど同じだっていうところがあって、さっとこういったものを作れたりというようなことが、あります。はい。で、もともとコンピュータービジョンをやっていたところから、創薬に来たっていうところで、なかなかちょっとそこの接続が分かりづらいところかと思いますので、もうちょっとそのあたりの話をできればと思います。で、ここにですね、顔画像が、いろいろとあるんですけれども、これ、すごい本物っぽい顔画像に見えますけれども、これ全部、偽物の顔画像、実在しない人の、顔画像なんですね。で、これはですね、もうちょっと、細かく言うと、ジェネレティブモデル、生成モデル、あとはその生成モデルの中にもいろいろ種類があるんですけど、特に GAN と呼ばれる、モデルで、生成された、顔画像ということに、なります。まあちょっとしたシワの感じとかいろんなところを見ると本当にすごいなっていうようなところですけど、こういったことができるようになってきているできるようになってきているというところが、あります。で、どうやってこういうことをやるんでしょうっていうところなんですけども、AI といいますか、機械学習と呼ばれている、ものはですね、データから学習するっていうアプローチになってますので、まず最初にやらないといけないこととしてはですね、顔画像を発生させる場合には、このような顔画像のデータセットを、用意しますと。で、その後ですね、AI のそのモデルに食わせて学習させて、で、それが完了すると、こういった顔画像が無尽蔵に出てくるというような、話です。で、これはこれで、非常に面白いアプリケーションがいろいろあったりするわけですけども、そうではなくてですね、例えばですけれども、低分子のデータセットを、使って同じようなことをやるとですね、今度、薬っぽい、ものが無尽蔵に出てくるっていうような、話になりますと。で、非常に、ポテンシャルがあるっていう、ところですと。で、当然、創薬はご存知の通り、非常にやっぱりその難しいですし、多目的最適化を行わなければならないですとか、非常に大変なことがたくさんあるので、当然こんなあの簡単に薬の候補が見つかるっていうわけではないですけれども、一方でその全然役に立たないわけでもないっていうようなところで、非常にそのポテンシャルが、ある、モデルになっておりますっていうところで、近年、AI 創薬非常に本当に話題になってます、盛り上がっていますけれども、このあたりがですね、盛り上がっている理由の一つなのではないかなと、思っております。で、この図はですね、僕らがどういった強みを持っているかっていうのを、表すのにもですね、非常にいい図なのかなと思っていまして、で、データの方で見ると、コンピュータービジョンの話と、あとは化合物を扱うような話って全然似ても似つかないみたいなところがあるんですけれども、一方ですね、モデルのアーキテクチャみたいなところで言うと、やっぱりその非常になんか背景技術は似ていたりするんですよね。で、僕らはモデルのところを突き詰めてずっとやってきていますので、ここは本当にどこにも負けないような技術を持っていると思っています。で、一方、製薬会社さんなんかは、非常にドメイン知識は僕らなんかよりも圧倒的にあるというところで、ここで組むと非常にシナジーがあるのではないかなというふうに思っております。で、次にですね、ちょっと、今なんか AI 創薬、盛り上がってきてはいるだろうけれども、実際にどんなものなのかっていうところを、ちょっとご紹介できればと思います。これ非常に面白い論文で、読んでみると良いのではないかなと思いますけれども、製薬会社、世界のその21社が、どんな活動状況になっているかっていうのを、まとめた論文になっています。で、左側の図はですね、2014年から18年の間で、その AI に関係したプロジェクトをなんかどれくらいやってるかっていうような、図になっています。で、 赤色のところではですね、自社のプロジェクトで、オレンジ色のところがなんかスタートアップと組んでやったりみたいな、ところです。で、これを見て分かることとしては、非常に力を入れて取り組んでいるところですと、自社内にその何個もプロジェクトあったりですとか、あとはスタートアップと組むっていうところも、一社と組んで終わりみたいなところではなくて、非常にいろんなところと組んでやっているっていうところが、あります。で、まだまだその数が少ないところもですね、今後どんどん数が増えてくるのではないかなというようなところで、ぜひ僕らとも一緒にご検討、僕らともお仕事していただければというふうに思いますという話です。で、あとは右側がですね、AI に関係する論文の数だったりしますと。アストラゼネカさんなんかは非常にアクティブに活動されているというような感じです。で、AI って言ってもですね、創薬の文脈で AI って言ってもですね、本当にいろんなところに使えるっていうことがあるんですよね。リンクコンソーシアムでも、プロジェクト30個ぐらいあったりしますし、AI 使う場所っていうのは、本当に非常に多様にあったりしますというところなんですけれども、特にどういうプロジェクトが多いかっていうのをこれを見ると分かりますということになっています。で、左側がドラッグディスカバリーで、右側がドラッグディベロップメントの話で、特にドラッグディスカバリーのところで言うと、コンパウンドっていうところがやっぱり非常に大きくなっていて、その中でも特にそのデザインとか、あとはプロパティ、これは分子設計したりとか、あとはプロパティ予測とかそういう話だと思うんですけども、そういったところがメインストリームになっていますというようなところです。これはデータセットとか、いろんな事情があると思うんですけれども、このあたりが非常にAI が適用されている部分になります。僕が論文をいろいろ見ている感じも、やっぱそういう感じなのかなという印象を受けるので、非常にそういう感じなのかなというような図です。で、あとはですね、これがですね、縦軸が各製薬会社のレベニューですね、トータルのレベニューで、右側がAI 関連論文の数です。各社が出している AI 関連の論文の数で、これって、AI 関連のR&D の予算ともこれ相関があるはずなので、どれぐらい AI に力を入れているかみたいなふうに読み替えることもできるのではないかなと思います。でちょっと若干分かりづらいですけれども、ここの線上にあるのが大体平均的なところで、緑の方ですね、右下の方に行くほど、AI に積極的に投資しているというようなところになっています。でだんだんですね、この右下のようなところに踏み込んでくる企業が、今後もっと増えていって、さらに AI すごく盛り上がっていくのではないかなというふうに思っております。はい。で、低分子のところで、低分子じゃなくてもですけれども、AI 創薬で登場するモデルって非常にたくさんあるわけですけれども、ざっとカテゴライズすると、この3つぐらいが非常によく登場するのかなと、思っています。で、1つ目がですね、プレディクティブモデル、予測モデルと呼ばれるもので、これは何かというとですね、入力が化合物で、出力がプロパティになりますというような、話です。でこれは非常にイメージがしやすいのではないかなと思います。で、シミュレーションと似てるんじゃないかみたいな話も、あるとは思うんですけれども、シミュレーションはですね、基本的に物理とか科学の法則に基づいて計算していきましょうというような話ですけれども、機械学習の場合はですね、データがあって、でそのデータからそのパターンを見出して、予測するっていうような、アルゴリズムに、なっていますというようなところです。で、一般的にですね、機械学習のモデルの方が、桁違いに速いというような特徴があったりしますというようなところです。はい、あとは、必ずしもシミュレーションと機械学習も相反するものではなくて、機械学習もデータがないと動かないので、シミュレーションで取ったデータを使うとか、そういった道なんかも場合によっては、あったりするというようなところです。で、2つ目がですね、生成モデル、ジェネレティブモデルですね。 これはですね、先ほどちょっとご紹介したようなモデルで、これはですね、モデルの出力がもう化合物っていうような、話になります。で、ちょうど、その一つ目のこの予測モデルとは、なんかその逆向きの関係に、あるようなモデルになっていますっていうようなところです。で、創薬って、よくよく考えると、基本的にはこの逆問題をもともと解きたかったんだと思うんですよね。やっぱりそのプロパティ得体っていうよりかは、その満たすべき条件が先にあって、で、そういう化合物ってどういうやつなんですかっていうところを解きたいっていうところだと思うんですけれども、化合物の探索スペースで非常に広いですと。で、いうところもあってですね、なかなかこういうことができなかったっていうところもあるんですけれども、近年、ジェネレーティブモデル、生成モデルが非常に発展してきたっていうところもあって、こういうことがだんだん本当に実際に役に立つレベルでできるようになってきたというようなところがあります。で、生成モデル非常に発展してきてるんですけれども、一方で課題がまだまだたくさんあるっていうことも、事実で、いろいろ課題あるんですけれども、その中でも多分一番大きい課題というか、注目されている課題としては、合成容易性、合成できるのかどうかが、分かんないっていうようなところです。で、そこでですね、逆合成解析モデルなんかも最近論文がどんどん出てくるようになってきているというところがあります。で、これを使うとですね、なんかその、新規のその化合物を得られたときに、それが合成可能なのかどうかっていうことを見たりですとか、あとは合成可能だとすれば、じゃあどういうルートで合成するのが最適なのかっていうのを出すモデルというふうになっています。で、今日のこの後のトークもですね、この流れに沿った形になっていまして、次のナジムの講演なんかは、これは新型コロナウイルスに関係する話ですけれども、基本的に予測モデルを使いましたっていう話になってますし、その後の順次の講演はですね、生成モデルの話で、最後、ロメオの講演が逆合成解析の話になっていますというようなところです。で、モデルですけれども、これらのモデル、ざっとどんな風に動くかみたいなところ、ちょっと別の見せ方でご説明できればと思います。で、生成モデルも、実は本当はなんかいろんなパターンがあるんですけれども、一つできることとしてはですね、まず、何もないところからそれらしい構造を出してくるっていうことができます。で、あとはですね、なんかもともとすでになんかその、なんか構造があって、なんかその、それを最適化したい。で、あとはなんかただの部分構造みたいなのでもいいんですけれども、何かしらのその構造があって、で、それを最適化したいみたいなこともできて、そういった使い道なんかも、あったりしますというようなところです。で、あとは、出てきた化合物のやっぱりプロパティ見たいっていうことも、あるので、予測モデルで、瞬時に予測したりですとか、あとはその合成経路を出すというような、話になっていたりしますというところです。で、これらの話、本当に細かいところまで入っていくとですね、なかなかキリがないんですけれども、僕のトークのところではですね、ざっとしたオーバービューみたいなところを話せればなと思います。はい。で、モデルについてこう考えていくときに、大事になってくるのはですね、レプレゼンテーション、その分子の表現方法なんですね。で、その表現方法とモデルのアーキテクチャっていうのは、非常に強く紐づいているので、この辺はよく考える必要が、あります。で、一つ目、よく使われるのはですね、皆さんご存知だと思いますけれども、フィンガープリントですと。で、フィンガープリントも非常にいろんなものがありますけれども、基本的にはですね、なんかある部分構造があったら1で、ある部分構造がなかったら0でみたいなことをすると、分子をこう01の塊で表すことができますと。で、01の塊に1回になっちゃう、要はデジタルな表現になっちゃえば、あとはそれをモデルに食わせて、ゴニョゴニョっと計算すると、なんかそのプロパティが予測できたりとか、そういった話になりますと。で、2つ目はですね、スマイルズです。で、スマイルズは化合物を、 文字列、こういった文字列で書き表すっていうような表現になっています。で、ですね、文字列になると何がいいかっていうことなんですけれども、機械学習 AI のそのモデルで、文字列扱うものってたくさんあるんですよね。自然言語処理とかそういう分野で。機械翻訳とかなんかそういうのもそうですし、AI になんか文章を書かせますみたいな話もそうですけれども、ああいったモデルが使えるようになりますと。で、AI にその文章を書かせる代わりに、スマイルズを書かせれば、なんかそれがなんか新しい化合物ですみたいな、そういう話です。で、本当かみたいなことですけども、意外とこれがうまく動くみたいなところがあったりしますと。で、あとですね、最後にその近年非常によく使われるようになってきたのが、グラフですね。で、グラフ構造っていうのはですね、こういったその点と線でこう表されるような構造をしていまして、で、化合物っていうのは、その原子と結合があってこうできているというふうにこう考えると、グラフ構造として表現するのが非常に自然に思えるというようなところになります。で、それぞれやっぱりですね、非常にそのメリット、デメリットが、ありますというようなところです。で、フィンガープリントの、大きなデメリットとしては、一つはインバーティブルでない、つまりそのフィンガープリントから化合物に戻せないんですね。化合物があったときにフィンガープリントにはできるんですけど、フィンガープリントから化合物に戻せないみたいなところが、起きますと。で、これ何が良くないかっていうとですね、特にその生成モデルっていう文脈で使うとですね、えっと、最後なんかその生成モデルからフィンガープリントが出てきて、でもなんか新しいフィンガープリント出てきたときに、これじゃあ結局どういう化合物なんだっけっていうのに戻せなくなってしまうと。で、実際どうやるかっていうと、この手法の場合には、例えばパブケムとかこう見に行って、なんか近い、なんか類似する化合物を拾ってくるみたいなことになるんですけども、そうするとですね、そのパブケムとかその既存のライブラリのなんか大きさによる制約を受けてしまうので、あんまり良くないみたいなところがあったりしますと。で、あとは、スマイルズですけれども、スマイルズの、課題みたいなところとしては、一つの化合物に対して1位に決まらないみたいなところもありますし、あとはですね、あんまりそのシミュラリティみたいなものを表現するようにデザインされてないんですね。で、例えばこの右上の図を見ていただきたいんですけれども、化合物、似ているものもですね、スマイルズとして表現すると、結構違ったものに見えてしまうっていうようなことが、あります。で、似ている化合物なのに、なんか全然違うスマイルズが来るってなると、それモデルにとっても学習しづらいはずなんですよね、普通に考えて。なので、そういった課題なんかがあったりはしますというところです。で、グラフはですね、非常にあの、近年よく使われるモデルで、グラフベースのモデルはですね、自動で特徴を抽出するみたいなアルゴリズムにこうなっていたりするんですけれども、これはこれでですね、全部が全部いいわけではなくて、グラフ系のモデル特有のちょっと厄介さみたいな話なんかもいろいろ出てきたりしますしっていうような事情がありますというところです。で、ちょっとだけ予測モデルの話をするとですね、で、今、3つほど表現方法をご紹介しましたし、えっと、で、どのモデルが一番強いかっていうのは、あの、タスクによって実は変わるんですね。グラフ系のモデルが一番新しいんですけども、必ずしもグラフ系のモデルが一番強いかっていうと、そうではなくて、タスクによって変わりますみたいなところが、あります。で、これはですね、17個の、異なるタスクで、それぞれどういったモデルが強かったかみたいなのを見てるっていうものなんですけれども、17個のうち11個のタスクではグラフ系のモデルが強かったですみたいな話になっていて、割とグラフ系のモデルが強い場合が多いんですけれども、絶対強いかどうかとは限らないみたいなところになります。で、次ですね、生成の話なんですけれども、なんか生成モデルって、こう、一口で言ってもですね、実は本当にめちゃめちゃいろいろバリエーションがありますっていうような、話に、なっています。で、なんでそんなバリエーションあるのかっていうところなんですけども、 いろいろですね、こうビルディングブロックがあって、それをいろいろこう組み合わせて、なんかモデルが成り立っているみたいなことに、生成モデルはなっているといいますか、機械学習のモデルって基本的にそうなっているってことなんですけれども、そういったそのビルディングブロックといいますか、パーツみたいなことを考えてもですね、この VAE とか GAN とか強化学習、あとはリカレントニューラルネットワークとかあってですね、でそれらをなんかこういかに組み合わせるかみたいなこう話があったりするので、非常にですね、モデルが多様になっているっていうところがあります。で、これはですね、去年の CBI でもご紹介した話ですけれども、なんかレビュー論文に載ってるこういったテーブルなんかを見るとですね、こんな風にですね、本当にいろんな論文、いろんな生成モデルが存在していて、で、それぞれできること、できないことってあったりするんですよね。例えばなんかなんか固定したなんか部分構造から出発してなんか構造を発生させたいみたいなことがあったときに、それをできるモデルとできないモデルがあったりするみたいなところがあるので、ちゃんとよく中身を理解して、利用していく必要が、あるのかなと、思っていますし、僕らはいろいろですね、こういったところで経験があるので、なんか、こういったところで僕らとなんか一緒にやりたいっていうところ、あの方がいるときには、ぜひお声掛けいただければなと、思います。で、ここからですね、ちょっとだけ事例みたいな紹介をできればと、思います。で、これは創薬というか材料の方の例にはなってしまうんですけれども、日本触媒さんとの事例ですと。で、日本触媒さんの方で反応性希釈剤っていうものを開発していてですね、これ工業用インクとかに混ぜられるものらしいんですけれども、開発していますと。で、ですね、法規制とかの問題で、この皮膚刺激性がもっと低いものを開発してくださいねというような、話がありますと。で、単純に皮膚刺激性が低いものを作ろうとするとですね、今度粘性が上がってしまうと、粘性も低くないといけないんですけれども、みたいなところが、ありますというところで、こういったところにですね、こういったことを探索するっていうときに、物理的に全部実験をやると非常に時間とお金がかかるということになってしまいますので、機械学習を使って、一緒にやりましたというようなところです。で、ですね、先ほどご紹介したような予測モデル、生成モデル、逆応性解析みたいなモデルを使ってですね、一緒に取り組んでいるというような形になります。で、あと関連することとしてはですね、これもマテリアルズインフォマティクスの方ですけれども、マテリアルズインフォマティクスのサービスを提供している会社として、グローバルでトップ20に選んでいただいたりということも最近ございましたというようなところです。で、ちょっと他の例としてはですね、予測モデルを使って、これは新型コロナウイルスで、ドラッグリポジショニングをやりましたっていうような話になっていてですね、これも一応アーカイブですけれども、論文化していて、あとは日経バイオテクさんですとか、そういったところに取り上げていただいたりというようなところが、ありました。で、この話についてはですね、この後、ナジムが話すことになっていますし、あとは、30日の口頭発表のところでもですね、話す予定になっているので、ぜひご参加いただければなと思います。であとは、最近発表したところとしてはですね、アステラス製薬さんと共同研究をしていまして、これも日経新聞ですとか、そういったものに取り上げていただいたりというところもあってですね、だんだん実績もついてきたのかなというふうに思っております。で、弊社ですね、それぞれどういった分野にどんなクライアントがいるのかみたいなところなんですけれども、コンピュータービジョンから創薬まで、このようなクライアントが、いますというような感じになっています。本当はクライアントもっとたくさんいるんですけれども、一応ロゴだったり名前を出す許可をもらっているので、ちょっと紹介できるところは限られてしまうんですけれども、こういったクライアントがいてやっていますというようなところです。特に AI 創薬は今年から非常に力を入れて営業しているみたいなところがあるので、ぜひ声掛けいただければなと思います。で最後になりますけれども、僕ら非常に変わっているところとしては、メンバーが、あると思っています。で、非常に多様なメンバーで、世界中からですね、こういった優秀なメンバーを集めてやっているっていうところが、あります。で、なかなかですね、日本語で仕事するっていうのは大変なことなので、思い切ってですね、会社の公用語を英語にして、それでですね、いろんな国から 来てくれたエンジニア研究者たちとコミュニケーションを取ってやってるっていうようなところが、あります。今は10カ国以上とかですね。で僕ら一番の強みはやっぱり AI ですので、AI に強い研究者、エンジニアがメインですけれども、やっぱりその創薬とか材料とかを扱うからにはですね、そういった分野のバックグラウンドを持つメンバーが非常に重要だっていうところで、ケミストリーとかバイオロジーのバックグラウンドを持つメンバーも最近増えてきていたりですとか、あとは、非常に研究開発に近いというか、研究開発をやっている領域なので、自然とその PhD 持ちが多かったりとかいうようなところになっています。はい。というところで、私のセッションは以上になりますというところです。はい。ちょっと質問が一つ来てますね。ジェネレーティブモデルは FBDD に適していますか？実施経験はありますかっていうような、ところですね。はい。これ、これフラグメントベースとかですかね。なんかJTVA とか、なんかそういったモデルはちょっと近いのかなと、思ってまして、そういったなんかフラグメントを組み合わせることによって生成するみたいなところがあって、でフラグメントになったとしても、非常にやっぱりその組み合わせの数って膨大ですので、非常に適していると、思います。はい、でそういったモデルなんかは僕らのところでも非常によく扱っていたりしますというようなところです。ちょっと質問の理解が合ってるといいなと思ってますけれども。はい。で、そうですね。あと、2つ目のご質問が、インプットの構造、グラフとかスマイルズによって生成モデルでも大きな差が出てくるのかみたいなところですね。で、これは、やっぱり差が出てきますね。で、これ、例えばなんか最適化なんかしようとしたときにも、その最適化したときに、なんかその、合成容易性が思いっきりそのなんか下がってしまいやすいとか、なんか変わりやすいとか、いろんなその特徴があるので、やっぱりモデルごとに結構特徴が違うっていうのはありますね。で、グラフ系のモデルは急速に最近発達してるっていうのがありますけれども、意外となんか昔からあるようなシンプルにこうスマイル生成するみたいなモデルが結構強いベースラインだったりしてみたいな話がいろいろとございますっていうようなところですね。はい。っていうところになります。はい。では、次はですね、次のセッション、次のトークに移りたいと思います。はい。Nazim, can you go next?
Sure. Okay, I'm sharing my screen. Please confirm that you can see my screen.
Yeah, I can see your slides.
Thank you. Okay. Hello, everyone. I'm Nazim. I would like to present you a case study that we did at Elix related to SARS-CoV-2 coronavirus. It's entitled Predicting Inhibitors for SARS-CoV-2 RNA-dependent RNA polymerase using machine learning. The talk will flow in the following way. At first, I'll introduce, perform general introduction and explain how we did target selection related to our study. I'll touch on data collection, the methodology we used, and I'll discuss our results. I would like to start with introducing SARS-CoV-2 coronavirus. During the start of the pandemic, we decided to use our tools and expertise to provide a contribution in the drug discovery against novel SARS coronavirus. At that time, the 3D structure of coronavirus was not resolved, and we decided to approach it approach this problem in a different way. At first, the quickest approach that we thought would be applicable was to do drug repurposing. For that reason, we decided to focus on RNA-dependent RNA polymerase. So RNA-dependent RNA polymerase is present in almost all viruses, all RNA viruses, excuse me. The reason, of course, lies in the critical role of RDRP in the replication cycle of RNA-based viruses. And since SARS-CoV-2 belongs to the family of RNA viruses, RDRP we chose RDRP to be our main target for the study. Related to the structure, despite minimal sequence similarity between various RDRPs of different RNA viruses, the structure remains relatively conserved between RDRPs of different viruses, and the active site especially conserved amino acids in the active site are present. So we performed multiple sequence alignments as well as structural alignments of the sequence of coronavirus RDRP to other viruses to make sure that the structural similarities are actually in place. And later on during our study, actually the 3D structure of RDRP were resolved and confirmed our analysis as well. So RDRP resembles a right hand with domains which are called as thumb, palm, and finger domains. Palm domain especially has a lot of conservation in terms of amino acids and charge particularly. And the domains of this particular RDRP, they span as usually in RNA viruses from A to G. And yes, it was confirmed that structurally SARS-CoV-2 coronavirus RDRP shares a lot of structural similarity to hepatitis C virus and poliovirus RDRPs, which was also in line with our previous preliminary analysis related to RDRP. One challenge I would like to mention was that when we started the study, of course, the 3D structure was not resolved, and it applies to many cases where, especially when novel viruses appear and it is difficult to solve the 3D structure of the target. So we decided to curate a data set of known RDRP inhibitors from the publicly available data and train machine learning models on the ligand data in order to see if we can train models to learn structural features in the ligands that can translate to their activity towards the RDRP enzymes. For that reason, we focused on filtering down and establishing a data set of RDRP inhibitors for which the activities were experimentally confirmed. And by activities, we focused on inhibitory dose and effective dose of the inhibitors. And we established a training set of 1,356 entries, 700 of which are active and 656 of which were inactive molecules. In addition to this data set, we established another set to test our models after training, which comprised 20 preclinical RDRP inhibitors, which were not, of course, included in the training set, and 20 unrelated, 20 inhibitors to the proteins of a distant family of proteins. And majority of them were kinase inhibitors. We had transporter inhibitors as well. So in this data set. And finally, we wanted to focus on FDA-approved drugs and drugs at the clinical stage in order to quickly screen them for the repurposing and find potential drug candidates that can be used for this particular problem. And we focused on antiviral drugs and anti-inflammatory drugs, and I will touch upon them later on during this presentation. Well, the main targets in our training set were hepatitis C virus RDRP inhibitors, dengue virus, poliovirus, and influenza virus RDRPs. Of course, this is the major limitation. Data is the major limitation in this case. So, and we further narrowed down to the experimentally confirmed inhibitors. That's why we ended up with this particular amount of entries for each particular RDRPs. And here I'd like to stress that we focus, of course, the values, the inhibitory activities are known for the particular enzyme, and they do not necessarily mean that the same activity will be applied to other enzymes, but nonetheless we thought that still we can try and see how the results look like. So I would like to stress this particular point in the data collection. Using this data set, we trained a set of classical machine learning models. We optimized them for this particular task and tested and found out that graph convolutional networks and random forest classifier trained on circular fingerprints and multi-layer perceptron of 3 layers trained on circular fingerprints performed the best in terms of area under the curve and accuracy. So we decided to use those 3 models out of the set to test them on the test sets that we prepared as well. So coming to the results. On the test sets, these 3 best models that we had performed relatively well. Of course, this is not a perfect result, but still we were able to see the trend and that the models learn the features from our training set, and were able to discriminate quite accurately RDRP inhibitors from other non-related molecules. And one thing we noticed was that for some models, for example, random forest performed quite well on discriminating true negatives. So only a small fraction of the molecules which were not inhibitors only could pass this filter. And we saw slightly different trends for each model, so that led us to think that the models might have learned in a different ways, and it might be useful to combine them in an ensemble model additionally to obtain better results. Indeed, when we performed the ensemble model, when we assembled these 3 models and performed not only 3 models, I would say, but when we established these ensemble models and evaluated the results, and the results on validation set were slightly improved according to our independent model results, and results on test set were also. Here you can see decrease in terms of area under the curve values and accuracy in test set compared to validation set. But of course, the validation set was tightly filtered, and our test set included quite molecules from sparse domains. So that might be one of the underlying reasons of the slightly lower performance on the test set. Nonetheless, we compared also evaluated how many true positives, true negatives, false positives, false negatives were detected in our test set compared to validation set. And basically, so results were not I wouldn't say that the results were perfect, but still we were able to see some effect, which indicated that our models learned how to learn the features related to RDRP inhibitors, and were able to discriminate RDRP inhibitors from non-related molecules. Coming to the results, when we analyzed our models, 4 models, 3 aforementioned our best performance and 1 ensemble model, we used those models to test them on the data set of drugs, approved drugs, and drugs in the clinical stage, to be able to identify some potential drug candidates for repurposing and narrow down the focus. So based on our results, interestingly, so we decided to compare results obtained by machine learning to a method that is commonly used generally for virtual screening. So we used open source tool AutoDocVina to screen the ligands, RDRP inhibitors, towards the result 3D structure of the SARS-CoV-2 RDRP, and compare the predictive binding energies by AutoDocVina, the standard tool quite often used, and by our models. So a predicted by number of models here, we indicate for each particular drug how many models classified it as a potential candidate against RDRP as an RDRP inhibitor. So beclabuvir and asunaprevir were identified by our models as potential candidates. Beclabuvir is indeed an RDRP inhibitor itself. It inhibits HCV RDRP, and it's non-nucleoside type of inhibitor. This was interesting for us to see. This indicated that all our models were able to identify or learn the properties of RDRPs and classify an actual RDRP as well, and suggest it as a potential candidate. And interestingly, we saw some number of HCV protease inhibitors being suggested by models as potential drug candidates, like asunaprevir, paritaprevir, and faldaprevir. And paritaprevir also shows quite high binding energy, low binding energy to the SARS-CoV-2 using AutoDocVina, and beclabuvir also shows some promising results here. And so I mentioned that we included some drugs at the clinical stage also, and remdesivir was one of the drugs, because at that point in time, it was preliminary results with remdesivir were out there, and we were curious if our models were able to identify remdesivir as well. And interestingly, we saw that our models learned to discriminate remdesivir, so identify remdesivir as well as a RDRP inhibitor, which actually was a positive indication of effectiveness of our models. In addition to antiviral drugs, since especially for patients with pneumonia, virus causes damage, and of course inflammation occurs, and antiviral drugs alone cannot help to relieve the inflammation. That's why we thought to look for anti-inflammatory drugs with potential activity against RDRPs predicted by our models and using AutoDocVina to see if we can identify potential candidates out of anti-inflammatory drugs that can be used also in this case, so that to alleviate inflammation in the patients with preceded coronavirus infection, and also tackle the virus itself. And interestingly, betoonilic acid and lupeol were predicted by 4 of our models to bear the properties of RDRP inhibitors, and so urusolic acid is also here. Interestingly, betoonilic acid and urusolic acid also are known to show antiviral properties, and particularly against HIV-1 type virus. So that was one of the interesting results. Of course, I need to mention that these are in silico predictions, and without actual proof by the experimental proof, it's difficult to say if these predictions are really correct or not, but at least we were able to see the tendency in terms of predicting and discriminating RDRP inhibitors out of the others, and at an acceptable accuracy. And identifying remdesivir was also one interesting finding that we obtained throughout our study. So to summarize what we did in this study, to tackle this problem, at first we had to establish a good data set. Using the open source data, we established a RDRP inhibitors data set for which the experimental activity values were established and evaluated. We filtered and cleaned up the data set to be used for training predictive models. And we compared our results of machine learning models compared to autodoc Vina predictions. And here I think it is important. It's important to know that autodoc Vina is and other docking tools are useful, of course, to do large screens, but drugs like remdesivir, for example, do not show promising binding energies there, but still they were they showed quite nice activities in this particular case. So of course they can, every tool has its pros and cons, and there are potential drug candidates that can be missed out. So we think by training machine learning models, we were able to capture that important information and suggest promising drug candidates. And remdesivir was predicted as a potential drug candidates along with other drugs and known RDRP inhibitors. And interestingly, we saw a cluster of protease, HCV protease inhibitors as potential drug candidates. And also comparing the machine learning models to autodoc Vina, we were able to see that the drugs, especially with the more confidence by machine learning models, had also quite low binding energy scores against SARS-CoV-2 RNA as calculated by autodoc Vina. So overall, the motivation of our study was to provide a contribution to the field and to be able to quickly suggest potential drug candidates to be tested and in vitro and in vivo, and to narrow down the focus on the potential drug inhibitors against SARS-CoV-2, and hopefully provide suggestions for good drug candidates. The results of the study we shared with the community, and we posted it on archive, so anyone can freely see the results and the details of our study. So with this, I would like to conclude my talk, and if you have further questions, please feel free to reach out to me on LinkedIn. Thank you for joining. And I would like to thank also Romeo, who will present later. So we did the study together with him, and he handled the technical parts of the study mostly. So thank you, Romeo, as well. And then I'll proceed to the questions. So the question is, how did you do curation of the training data? Thank you. So I would like to take you to this slide. Yeah, we screened PubChem and Campbell Bio assays for RNA polymerases, and then narrowed down the molecules to RDRP inhibitors of various viruses. Then we ended up with a quite large data set, but then we narrowed it down further. So we made sure that no DNA polymerase inhibitors were in the data set, and then we narrowed down to the inhibitors that were actually confirmed in vitro, at least in vitro assays, and for which the IC50 and EC50 values were known. So this left us with 1,356 entries. And as you might anticipate, that the IC50 and EC50 values were quite dispersed between the different inhibitors. So we tried different cutoff thresholds for the IC50 and EC50 value. Here we decided to kind of merge those two values and use them together, because for some molecules we knew only IC50 values, for the others EC50 values, and in order to increase the number of the molecules within the data set, we decided to do that way. And we experimented with different cutoff thresholds, and finally we set the cutoff threshold between active and inactive molecules to 5 micromolar, because from the publicly available data, when we curated it, so there were molecules also like they were labeled with the activity either active or inactive, but for some molecules they were labeled active even if IC50 values are quite high. So we decided to standardize it and set up a cutoff threshold. And we finally decided to use 5 micromolar as a cutoff threshold of IC50 and EC50 values, and that gave us promising results actually. And thank you for the question. And with this, I think I can conclude this talk.
Yeah. And thank you so much, Nazim.
You're welcome.
So Junjin, can you go next?
Sure. Okay. Can you see my slides?
Yes.
Yeah, I can see your slides now.
Okay. Thank you. Good afternoon, everyone. My name is Junjin, and today I'd like to present on behalf of Elix on the generative models. So I'd like to begin talking about generative models for molecular design. In today's standard, trying to find new molecules is fundamentally difficult, but we all know that computers are pretty good at it. So, trying to explore the space of trying to figure out to discover a new molecule is extremely difficult. But most likely we would like to find new types of polymers or new types of drugs. And we know that exploring this landscape is tedious and meticulous because it's repetitive. Sometimes drugs work, sometimes it doesn't. And each exploration that we see here is a cost to the chemist himself who needs to basically synthesize this in the real world. So one solution to look into drug synthesis is to virtually do it. And in this case, we are looking at generative models. One way to solve this problem is to basically use machine learning. And why machine learning? Because machine learning are pretty good at local optimization. If we see this picture right on our left here, we have a landscape of our chemical space. All we need to do is basically minimize our search by going down this hill in such a way that the lowest peak at this landscape is basically what we are looking for. In reality, by minimizing this landscape or traversing this through this chemical space or any space that we are interested in, we can basically try to sample from this space and generate something new. For instance, in the image domain, if we traverse through this space and we find the local minima and we try to sample anything within the space, we would be able to construct some kind of realistic images. One of the most popular methods so far has been proposed by NVIDIA called StyleGAN. And what this means is that simply sampling from this valley right here, we are able to generate some kind of new images that are very realistic-like. Likewise, if we try to do it for chemistry or basically molecules, we can find something very interesting. For instance, this person here, Aditya Joshi, has actually successfully taught a simple model, not a simple model, but a very complex model called GPT-3, which was recently proposed by OpenAI. And just training it with just 4 examples, it's able to tell us whether this prediction is combustible or otherwise. So this tells us that machine learning methods are very good at learning some kind of local optimization, and it can recognize patterns very quickly. So rather than trying to scrape through all our landscape, our infinite landscape of chemical space, we are more excited to find a method that minimizes this local optima. This brings me to the problem definition of generative models in the molecule design team. Basically, given the task of trying to find a new molecule, we are presented with a very large search space. The goal here is to minimize this search space in such a way that we do not require exorbitantly years or thousands of computational hours so that we can learn how to generate new molecules. What this means here is that we are proposed with several challenges. We know that we have this search space, but how do we really construct this generative model in such a way that we can basically sample from it to get back our molecules? This brings us to several questions. Initially, there were challenges in this field, namely, how do we assemble candidate compounds? We know that we have molecules, but how do we bring it down to the level that a machine can understand and try to extrapolate from this particular search space? Next, we know, let's say we are capable of sampling from this search space. How do we actually evaluate the potential quality of these molecules? This is also a very challenging task. Finally, if we are able to sample from this search space, how do we actually sample from it effectively? Sampling just from a small local optima usually gives us a very similar-like molecule, but doesn't mean that it's novel. Depending on the constraint that we're interested in, this becomes a very difficult task. So one likely solution so far, probably you have seen right now, is to propose what we call a deep generative model. So deep generative models are some kind of models that utilizes deep learning or a neural network to discover a new molecule. What are they exactly? In short, if I give you a pictorial here, we have a molecule of the following. All we need to do is translate it into a machine learnable representation. Let's assume that this representation here is a vector of matrix or some kind of a machine code that we can then put it into our encoder. And what we want this encoder here is to bring us down into this latent space. Finally, from this latent space, all we need to do is sample from it and put it in through a decoder so that we can obtain back our molecule. Now, what we are interested in is actually the latent space itself. This latent space here is what we are going to search and what we are going to be able to sample from. How do we sample? We are basically constructing this latent space as a probability distribution. So by sampling from here, we can then generate a new molecule. So this architecture here is basically what we call encoder-decoder architecture or decoder-encoder architecture of interest. And what we want here is basically to tell a machine or our computer to learn automatically. So the input and the output are equal. And what we want to do is minimize the loss from the output to the input so that we can learn the probability distribution. Now, what we have, since we have this probability distribution, we can sample from it and we can generate new kinds of molecules. There are several challenges in molecule design, but particularly one problem is how do we actually train it to learn automatically? One way to do this is by treating everything as a representation. What this means is that, let's say you have a 2D molecule. We can then represent this as smile strings. And a smile string is basically a string of characters concatenated together. And from these smile strings, we like to bring it down into a vector space or a vector so that the machine can understand what this means. Usually this vector here can be forms of a fingerprint or otherwise one hot encoder. What this means is that the entire vector here can be zeros and ones or maybe some kind of real number. We then put this vector into our encoder. Usually it's a set of vectors, which means it's a matrix. Batches of them would be plugged into the encoder, and then we would go through this same process of bringing it into a latent space, and from the latent space, sample it and bring it back into the decoder, and finally we get back our representation. This purpose of doing this is so that we can utilize recent advances in machine learning tools such as PyTorch or TensorFlow that allows us to do automatic differentiation. What this means is that from just computing this model, we can then backpropagate our loss to the encoder and train it bit by bit through gradient descent. And this would give us a much, this would give us a much more interpretable model that we can then sample from. The other alternative here is to introduce something called a graph-based model. In this slide, I talked about smile strings, but there's another way to represent molecules in terms of graph. And one way to do this is basically take the same 2D structure. We would then present it in a graph form, let's say like this. And if you look at this graph structure, it's very similar to the original 2D structure here. Then we transform this graph structure into a adjacency matrix. Now we have a adjacency matrix to tell us which nodes are neighbors to one another. We also have each of these nodes having their own features. Let's say maybe this node here might have some kind of unique features of some kind of property that is unique to this particular node. And from there, all we need to do is take this node features and adjacency matrix, either through some kind of concatenation or multiplication process, and put it into our autoencoder. The resultant output then would be our agency matrix, and this would then be reconstructed back into our 2D molecule. This process is ideal in most cases, and graph structures are usually chosen because it's more faithful to the representation of 2D molecules. Small strings, on the other hand, are more cumbersome. At times, if you manipulate the small strings in different order, it would break the representation of what we're trying to say from the 2D structure. Hence, lately, there has been a rise in number of works related to graph-based autoencoders. Initially, I've talked about this, so I'd like to explain a bit more about the encoding part. So what we have initially was a graph structure. The idea of how we embed this graph here into a vector is by a method called message passing. All we need to do, care is that if we initialize this graph right here, and we have some kind of information centered around here, all we need to do is figure out how do we pass on information to our neighbors, and then aggregate it from our neighbors and bring it down into this central node here. This central node here would then have information from all its neighbors, and then what we want to have here is that this representation would be similar to its neighbors. Ideally, this representation would tell us nodes that are similar to one another would give us very similar properties or some kind of hidden latent code that we cannot hand code. And thus, from this latent code, we would like to sum everything together and form it into a matrix. So by using a graph encoder, or we can swap out a different type of encoder, we can then basically flexibly change what type of inputs we can give. In this case, graph encoders are more useful because graph encoders can encode additional informations like features because from atoms in this case, whereas small strings are usually more cumbersome, and hence there are various ways to do this. So fundamentally, there are pros and cons from using small strings versus a graph-based method. Now, to take a look at our in-house task, usually we have clients asking, if we are about to find a new molecule, can we actually specify types of properties of interest? So our answer usually would be yes, we can basically propose a model for you, and usually we can try to preserve these properties. What this means is that instead of just trying to generate a simple molecule from this generative model, we sometimes need to constrain the space of our latent space to ensure that certain properties are preserved. So sometimes we may add an additional neural network, for instance, right here, and we would like to ensure that this property here is minimized from the loss. So this inclusion of constraints would then help us to restrain this search space. Ideally, we have only one type of properties, but in the real world, we would have many types of properties, and this could be a multi-objective minimization problem, which is a whole new level of a problem that we need to solve. And yeah. So this brings me to the challenges currently that we face as a startup company in Elix. The question usually we ask ourselves is, how can we build generative models that scale? Right now, most of the generative models that are proposed are computationally expensive, and they are only limited to very small molecules. The reasoning behind this is because if we use graph-based models, the matrix of each molecules are right now are very small. If we try to enlarge it, we would have scalability issues. There are some fundamental ways to circumvent this, but it's really in another domain called the graph learning, representation learning problem. So the next thing we ask ourselves is, if we can build a generative model, can we ensure that this model here that we built would allow us to generate a synthesizable molecule? This is a difficult question that we are usually consulting ourselves. Sometimes we have a database of all molecules, and all these molecules here are synthesizable by default. So we expect our generator to learn from this database in such a way that all the molecules that are generated would then be synthesizable. In most cases, this is not true. So we may need to add additional filters, like a post hoc filter, to say that this generated molecule either fails or do not. Likewise, this approach here requires some kind of prior information, and it's usually not ideal because we are not chemists sometimes, and sometimes we need to have some kind of rules, and this kind of rules are hard to come by. Which brings us to our next problem. Even though we have a generative model like this, we can predict some kind or generate some kind of new molecule. It's not straightforward to tell us how can we take from step A to step Z. In between is basically a black box, and hence one open question here is, how can we dissect this generative model to ensure that we're understanding or basically able to interpret what this sampling process is. The last thing that we are currently facing as a challenge here is about validity and stability. In most cases, models here are very simplistic, especially from the research perspective. Trying to include more constraints like prices or yields, this is very difficult. In most cases, data such as prices and yields are absent. And how can we make use of this little information that is partly usually confidential to each pharmaceutical company? So this is a challenging problem. It also brings us to having bias and variance problem. As we try to encode more information or more constraint into our model, we would then face off with a problem of the generalizability. Will our model start to learn how to generalize better, or will it just start to memorize the data given? And yeah, so this summarizes some of the challenges that we're facing from a generative model. And in Elix, we are keen to look at research like this, and we are pursuing this type of problems. So just to summarize, the state of the current generative models can be summarized into several streams. Mostly, some of you may have known, generative adversarial networks, reinforcement learning, recurrent neural networks, and variational autoencoders. In recent state of work, there are different types of ways to come about this. Some use smile strings, some use graph-based approach. But there are still some problems that we need to consider fundamentally from a machine learning perspective. For example, if we can shrink this chemical space further, then we are able to basically interpolate and generate some kind of new molecule in a more viable manner. Maybe this molecule here would be more valid and more synthesizable. There are some approaches to this, and some of them include semi-supervised learning, active learning, and meta-learning. All these approaches here are some ways to optimize our learning approach, and hence we are trying to actively explore different ways to do this. On the other hand, some methods like VAE or let's say reinforcement learning is not really interpretable. At every time step we take, or every time we do a training on the VAE, for instance, we are not sure why the model generates molecules like this. So there needs to be a way that tells us that each of this search space here can be separated into different kinds of properties, different types of features, and so forth. Recently, there have been work in normalizing flows, trying to basically reshape this probability distribution in such a way that we are able to sample from a certain peak that would tell us that this type of features are encoded in our molecule. There are other ways to do this too, like disentangle VAE and so forth. It's still an active area of research that hasn't been quite applied to molecule design, drug discovery, for instance, but have been actively explored in other domains such as computer vision. And last but not least, current state of the work are kind of limited to smiles and graphs only. What if there are better ways to represent the input? For instance, right now we look at 2D structures and then we convert them into smiles or graphs. This conversion process usually gives us a loss of information. And for usually what we want is a more ground truth like input, rather than trying to simplify this input too much in such a way we lose information. So one way to look at this is to see if we can include dynamics or take 3D images, for instance, and this would give us more information, and it might even help us to create better molecules. So this brings us to another problem. If you're trying to simulate a real-world experience with drugs, it's also not so straightforward. Sometimes doing it virtually means that it's only viable in virtual, but what about real-world experiments? Does it really work? And sometimes the feedback loop is not ideal. Virtually, it's less expensive compared to asking a chemist to synthesize it each time and give us a feedback whether this molecule works or otherwise. There's also a problem of trying to understand the theoretical interpolations of the latent space. We know that we are sampling from it, but how do we ensure that this bounds between this latent space is what we want to sample? So for instance, in this work here by Bradshaw, we can simplify this approach by taking a multi-model approach. So likewise, it's an autoencoder architecture. But in this encoder architecture here, we're taking a graph variant, and in the decoder, we're taking a RNN approach. So given two types of molecules, let's say the reactants, right here, we are trying to embed it into a latent space using graph neural networks or some kind of message passing algorithm. From the latent space, we are then interested to sample from here and put it into our decoder in such a way that we can then obtain back the set of similar reactants. This process of getting a reactants and then trying to predict the reactions from the reactants to products is one way to simulate a real-world experience, because if we only do from this reactions to the products in a single step, in most cases we're not sure why what are the in-between steps that we need to take to get to this final product. So there are various challenges in generative models, and these are some of the things that we are looking at Elix and trying to improve them. This brings me to the conclusion of my talk. At Elix, we try to explore various ways to improve generative models. Particularly, we look at our client needs, and one of them could be mostly related to property constraints or particular properties that we would like to encode into our molecule. Alternatively, we also need to look at molecules, ways to generate molecules that are synthesizable. So this really depends on our client needs. Additionally, we also conduct independent research to improve our in-house generative models platform. And this could be related to trying to improve the encoder or the decoder, or some kind of introducing some kind of new optimization approach into our model. So just to summarize, generative models for molecular design is an active area of research. There are still many fundamental problems that are unexplored, and these problems are well known in some of the machine learning arena, but how applicable it is to this molecular drug discovery problem is still quite new, and it requires further analysis as well as further understanding. And I would like to also say that most methods right now are also data-driven, and they're kind of unsupervised. Sometimes having an additional prior knowledge would help to improve the models, but how do we encode this prior knowledge in such a way that we do not tell the model to memorize or depend too much on this prior knowledge? We only want it to know that this is one way to start, and we would like it to then further discover other ways to interpolate and generate molecules. So another big question is, can we actually avoid templates altogether and so forth? So this concludes my talk. If there's any questions, please feel free to ask. These are the references that I've included in my presentation. Thank you very much.
Thank you so much, Junjin. Can you answer the question by text? Yeah.
So, okay.
Yeah. And I will translate for the Japanese question.
Okay.
Yeah.
So the question by Koichi-san about the Bayesian optimization. Yes, there has been a work on related to this, and one of them has actually tried to use Bayesian optimization to go through this search space. And I think the limitation of this Bayesian approach is not about the novelty or whatever compound that was found, but rather it was very computationally expensive. So it was not a very ideal solution for larger molecules. It works for very small molecules, and yeah, it can produce quite novel and quite valid compound.
Actually, the first question is about patent. And is it possible to generate molecules which are not patented?
Right. Trying to generate molecules that are not patented is our goal, because our data sets are mostly composed of data that we scrape from patents like USPTO, for instance. And then we try to teach the model that you have to generate a molecule using this rules or this set of training data that we have. So ideally, what we would like is to be able to generate molecules that are not in the patent. I hope that answers the question.
Thank you so much.
Thank you.
So, okay, so let's move to the next presentation. So can you start, Romeo?
Yes. So hello, and thank you for attending. I'm Romeo Cozac. I'm a deep learning specialist focused on chemistry and especially drug discovery at Elix. Today, I would like to talk about retrosynthetic route prediction and how AI can be leveraged to solve such a challenging problem. It is a challenging problem indeed. The space of possible reactions is vast. Reactants often contain multiple reactive centers, and oftentimes the presence of a certain functional group might make the obvious choice invalid. But even if we solved all these problems, to have a useful, production-ready system, this is not enough. It is very unlikely that a single decomposition of a desired product would yield a commercially available reactant. It is not uncommon to have chains of 10 reactions or more, and one end product can often be obtained in multiple ways. We then need to decide which routes make more sense, which starting materials are cheaper, and which paths result in higher yields. But to perform multi-step retrosynthetic route prediction, we first need to solve single-step prediction. The machine learning approaches applied to reaction prediction can generally be classified as either template-based or template-free. Template-based approaches start by extracting a set of reaction rules, which are then used to classify reactions and to perform in silico reaction simulations. One common strategy is to extract the reaction center, which basically represents the set of atoms and bonds which change during the reaction, and the reaction center's next adjacent neighbors. So taking a look at this example, we see that atom marked as 1 here changes during the reaction as it forms a new bond. And in the second reactant, carbon number 2 and the oxygen changes, which again, the double bond becomes a single bond. So in the final rule, these 3 atoms would be included along with its next adjacent atoms, which happens to be a carbon atom in both cases. If our desired end product matches any of these extracted templates, we can apply them to obtain the starting materials, the reactants. But it's not that easy. Even though we can now simulate reactions, a rule is not enough to reliably predict them because they ignore the global context of the whole molecule and they only focus on the reaction center. In this example given by Marvin Seidler and colleagues, we can see that multiple reaction rules can be applied to the desired target, namely the Suzuki coupling and the Kumada coupling reactions. But the aldehyde in this context conflicts with the Grignard reagent, and so Kumada coupling would probably not work very well. As we can see, not all applicable reaction rules are valid. Other times, multiple rules could be valid, but one of them might overshadow the others in terms of yield/purity. To overcome this setback, we could train a neural network to predict which rules are more likely to apply in a certain context. In this sense, the input of the model would be the desired end product, or to be more accurate, a feature-as representation of it, like maybe a topological fingerprint, and the output would be the reaction class. To be more specific, the output is a probability distribution, and each template would have a certain probability that we can sort of think of as confidence. In this example, the model allocates most of its confidence to the Suzuki reactions, and almost none to Kumada coupling. And we can use this confidence not only to get the most likely prediction, but a ranking of the top K templates that are most likely to apply. With almost 9,000 reaction templates, this model can achieve a top 1 accuracy of around 64% and a top 10 accuracy of 95%, which is quite impressive. But not all that glitters is gold. While reaction templates have a high validity and models trained on them have good accuracies, our search field is limited to the reaction rules we extract. The models won't be able to predict less common reactions for which we have no templates. Plus, we have to consider data availability. For the above experiment with these 9,000 templates I just mentioned, the dataset contained at least 100 reactions for each and every template. In this case, there aren't enough reactions for a certain template. It is excluded and the data remains unused. Of course, we could always lower the threshold and allow templates with as little as 50 or 10 reactions to be part of the data set, but the accuracy is expected to decrease because we have more reaction classes to consider and fewer data points to train on. But there are ways to combat these problems. One approach is pre-training with augmented data. In this approach I am describing, the authors created a dataset by applying all the extracted templates to all the available reactions in the dataset. If the generated reactants match the real data, the entry is used to pre-train the model. Then during the fine-tuning phase, the model is trained on the real data with the true reaction template. Intuitively speaking, the model first learns which templates work in a given context, then it learns which rules are the best ones for the current job. This strategy is very helpful, especially for low occurrence templates, since the model will be able to explore them in more detail. Another approach, of course, is to use a template-free model. And template-free models, as the name suggests, avoid reaction templates altogether and try to predict the reactants directly. Template-free models can come at the cost of reduced accuracy, but we gain a lot of flexibility and we can avoid the limitations of reaction templates. But even template-free models have requirements. If the data is sparsely available for certain types of reactions, the performance will suffer in those areas, even for template-free models. Maybe the best way forward would be to combine these two approaches and get the best from both worlds. Anyway, as an example of template-free model, I'm presenting here a paper called Learning Graph Models for Template-Free Retrosynthesis, in which the authors use a graph-based approach to solve retrosynthetic root prediction. Graphs are, as my colleagues mentioned, data structures composed of nodes and interconnected through edges. In the case of molecules, we can think of the atoms as nodes and the bonds between them as the edges. And each node will have a set of features, like a one-hot encoding of the chemical symbol, maybe the valency, the degree of the node, maybe whether it is part of a ring or not. It can be anything we find useful. These features are then passed through graph convolutional layers, which aggregate the information from further away nodes. This process mentioned in the paper can be divided into two steps. First, the model learns using a graph-based network, a distribution of graph edits, like breaking a bond at a certain location, as pictured here. By performing this operation, we end up with several syntons, which are essentially fragments or incomplete reactants. In the second phase, another model is trained to predict fragments that complete these syntons, which are then concatenated, and they form the final reactants. Other graph-based methods are popular, but a different approach to solving this problem is using sequence models. Maybe I won't go into too many details as the time is a bit short, but just as a short summary, in the past, recurrent networks would be used for this task, which basically would get the input sequence and use each one of them one by one. And for example, the first input would be encoded, tokenized and encoded, and it would be fed to the second input along with its own encoding. This process is done until we exhaust the whole sequence, after which the decoding phase starts. And in the decoding phase, the first token would receive as input the least predicted node, or the rest predicted token, and predict the first token as output. Then the output of this is used in the second for the second token, and so on and so forth. In terms of reaction prediction, we can think of the encoder part as the end product, or in case of forward prediction, we can think of them as the reactants and regions, and the decoder is the end product. And there are more novel approaches like transformers, which unfortunately I don't have time to cover right now. But of course, there's more to reaction prediction than just focusing on products and reactants. There's a question of what is the context of a given reaction? It is not enough to just focus on the reactants. Oftentimes, catalysts and solvents are very important for the success of the reaction, including the temperature and the pressure and other reaction conditions. There are approaches which can help us predict these values, but there are still many unanswered questions left. For example, what is the yield? What is the purity of the end result? Whether there are byproducts, and we only covered single-step reaction prediction. Multi-step prediction is a whole different beast. Unfortunately, I don't have time to cover all these topics, but if you'd be interested in finding out more, please reach out on LinkedIn. I would be happy to have a chat and offer you advice the best way I can. Thank you.
Thank you so much, Romeo. はい、ということで、エリックスのですね、トークはこれで以上となります。でもしご質問だったりですね、僕らともし一緒にご仕事、一緒に仕事をするっていうことを検討していただけるみたいなことがございましたらですね、会社のですね、お問い合わせフォームにご連絡いただくとですね、僕常に見ていますので、はい、何かしら、質問でしたり、何かしら共同研究ですとかご連絡いただければと思います。はい。ということで、以上になります。はい。少し時間オーバーしてしまいましたけれども、多くの皆さんにご参加いただいて非常に嬉しく思っております。どうもありがとうございました。以上になります。